here. Thank you. Let me introduce our speaker for this afternoon. Craig Benham received his PhD in 1972 from Princeton. He became interested in protein structure problems while an assistant professor at Notre Dame, after which he spent a year working as a postdoc in the biology division at Caltech. A few of his recent papers include The Theory of DNA Superhelicity, Writhing and Linking Densities for Closed Circular DNA, and my personal favorite, of Chicken Teeth and Mouse Eyes, or Generalized Character Compatibility. He's a member of the mathematics faculty, the biomedical engineering graduate group, and the Genome Center at UC Davis. Before coming to UC Davis, he held positions at the University of Notre Dame, Lawrence University, the University of Kentucky, and the Mount Sinai School of Medicine. His research group at UC Davis has developed statistical mechanical methods to analyze computationally the occurrence of structural transitions in stressed DNA molecules. I'm very pleased he was able to speak with us today. Please join me in welcoming Professor Benham.
Well, thank you, Abby. I appreciate being here. As I was telling Abby before, I went to a very similar program myself when I was in, when I was in high school many, many years ago, and it really set the course of my career. I think these programs are wonderful and very important, and I hope you're all really enjoying it. The program I went to was in a single field at the time, unlike this where there are clusters who are working in any of perhaps 10 different areas. I wanted to talk today about DNA, which is, as you can tell, my first love, scientific love. And, but I wanted to show you a problem, an area where a lot of the fields that you're studying separately come together. I mean, it's natural when you need to learn physics, chemistry to learn chemistry, biology to learn biology. But when you get out into the real world of scientific research, it quite often happens that a particular problem has aspects that are biological, aspects that are chemical, aspects that are physical, aspects where you need mathematics to understand them. So all of these things in real world problems very often come together. And I wanted to talk today to give you an example of this. So the mathematics that the DNA uses is called topology. I'm going to give you a little introduction to what that means and what it is as it's relevant to DNA. And then I'm going to talk about how DNA uses topology to do the various jobs that it does. And I hope I'll be able to end with a particular example of this. I want this talk to be relatively non-technical. And also I want you to see where all these different scientific areas contribute to it. But if there are any questions or problems, don't hesitate to raise your hand. Okay, so topology is basically, it's the study of the properties of shapes that don't change when you change the shape. That is, you can continuously deform this coffee cup into a donut. The shape of a donut, or the shape of a coffee cup, is its geometry. The topology that doesn't change is basically that it has this handle, which stays there no matter what you do. So as long as you're continuously deforming something, you're not changing the topology. Now, what we're going to be working with is DNA, which is a molecule that's a linear molecule. So we're going to be interested in linear things mostly. So here is a straight line, and here is a deformed version of a straight line. These two, of course, have the same topology. To get from this one to that one, you just continuously deform it like a belt, a line segment. You know, no matter what I do to it, it's still got the topology of a line segment. And the essential thing that doesn't change is that it's got two ends. This is different than the topology of a circle. See, at these ends, At any interior point, there are points on either side of that point. At the ends, there's only points in one side, but not the other. And similarly for that end. But for all of these points, there's a point on either side of them. So it doesn't have ends. And these two are topologically different. The things that are forbidden are you cannot cut something. That's a discontinuous change. You cannot glue two things together. I cannot glue the ends of these two points together to make sort of one point out of the two. Because that's a discontinuous change. But anything that I want to do, wrap it up and snarl it up as much as I want, preserves the topology. And for instance, if I close this, then it's the same deal. I can just wrap it and snarl it, but it's still, whatever the geometric shape is, it has the topological structure of a circle. Okay? So, knots. When you think of a knot, you informally think of, you know, tying a knot in your shoe or something like this. That, from the point of view of a mathematician, is not a knot because I made it by a continuous deformation of this line, and I can get rid of it by a continuous deformation, and it's just still the same line. But to make it a topological thing, if I close it up, so that now it's a knot in a closed loop, then that knot is stuck. There it is. There's nothing I can do to get rid of it. I can pull it tight, I can move it around, I can mess with it all kinds of different ways so that it's not apparent that it's a knot, but it still is. It still has this knot in it. So this knot is a topological thing once it is in a closed circle. Okay? And there are lots of different knots. There are whole tables of them. These are the one that I put in here is the simplest of them, a trefoil knot. This is a granny knot. There are tons and tons of others. The cousin to a knot is what's called a link. To make a link, you need two closed circles, and they can link through each other in a variety of ways. This sort of thing is also a topological property. As long as the two are closed, you cannot unlink these unless you cut one and allow it to pass through the other and then close it up on the other side. So that's a discontinuous change, so that's not permitted. So these guys are going to stay linked no matter how you mess with their geometry. And again, as with knots, so with links, there are tons and tons of different ones. There are tons of different ways you can interlink two circles. And these are topologically, each of them distinct. This is a very wonderful field to study as a field of mathematics. In some ways it's quite intuitive, in some ways it's very hands-on, and in some ways it's very abstract and uses a lot of tools of abstract mathematics. So it's a wonderful first introduction to abstract mathematics. Now, in topology, one of the things that people do is they find invariance. Some number that doesn't change as long as the topology stays the same. And the linking number is one of them. It's the one that's relevant to this talk. The linking number tells you the number of times one closed circle links through the other. So in this case, if you follow the blue circle, it links from below to above through the red circle. And you call that you give that a +1. And if it linked the other way, you could give it a -1. Here, the blue circle links from below to above there, and then again from below to above there, linking through the red circle. This piece is outside the red circle, so it doesn't contribute. So it's only linking from below to above through the red circle that matters. So this guy has linking number 2. Okay? And it turns out that you can show that the linking number is a topological invariant. No matter how you change the geometry of this, whatever structure you have will still have a linking number 2. And there are a lot of other ways to calculate linking numbers. I just want to give you an understanding of kind of what they mean and a simple way to visualize them. So for example, with this guy, this belt has a white side and a black side, so we don't get confused. So I'd make one turn and 2 turns, and now I can close it up. And now if you think about the 2 edges of the belt as being the 2 circles that are interlinked, you can see that there are 2 full turns there. So one edge is linking through the other one twice. Either one you look at, it's linking through the other one twice. Can people see these things? Okay, good, good. So this has linking number 2. But again, you know, it has all kinds of different geometries it can go into. But it still always has linking number 2. And it's unknotted. Okay, so this one, again, the blue goes through the red from below to above there once, and there once, and there once, and there once. And so this one has linking number 4. And this, I hope, is starting to look a little like DNA. So let me take a moment and talk about DNA. So we'll go from the mathematics to the biology here for a moment. DNA structure. This is the sort of standard structure of DNA, but DNA structure is basically there are 2 strands, a red strand and a blue strand, just to keep them separate, you know, in your mind. They are held together by weak bonds. These little dashed lines here are hydrogen bonds. They are weak bonds. They're basically electrostatic associations that opposite charges attract. So they're not hard chemical bonds, not covalent bonds. They're just, so you can easily separate these 2 strands by just pulling apart these weak bonds. Each strand consists of a linear array of repeated objects. The objects are a base, which has some nitrogens in it, and a sugar. In this case, the sugar is deoxyribose, but that doesn't matter. And a phosphate group. The phosphate groups link 2 sugars. So the one piece is connected to the next by the phosphate group. So it's a base, a sugar, and the phosphate group. Now the sugar has the carbons are numbered 1, 2, 3, 4, and 5. So the sugar connects through its 5' carbon. It connects to one phosphate. Through its 3' carbon, it connects to the other phosphate. So there's a 5' to 3' chemical direction that goes up and is preserved in this. Now, all this is kind of for the chemists, but the point for everybody else is just that there is a directionality to this which gives an arrow to the directionality of the strand of the DNA, and that the other strand has the opposite directionality, the 5' end. Here, this sugar, this is the 5' carbon, this is the 3' carbon. So 5' to 3' is down in this one. So that one is going down and this guy's going up. Okay? The thing that people most associate with DNA is that it has a base sequence. That is, these bases can be any of 4 things, call them A, C, T, or G, and they base pair across. The bases on one strand pair with the bases on the other, and it's always C with G or A with T. Okay. So each strand contains the information to make the other strand. If you take away the blue strand and you just have the red strand left, you can make the blue strand again by, well, here's a T, so I have to put an A across from it. Here's a G, I have to put a C across from it. There's an A, so I need a T over there. Here's a C, so I put a G over there. So each one of them contains the information to make the whole molecule. And also, of course, as you go along the DNA, you read, there's a sequence of bases, and that's how the genetic information is contained. And then, of course, at the end, the whole thing is arranged as a helix. Okay. Now, the topology comes in because DNA also initially came in because DNA also was found to be occurring as a circle. And this one shows it best. These are the original electron microscope pictures from the original paper that discovered this, which was 10 years after the double helix by Watson and Crick was published. So now we have a circular molecule that has a linking number, because it's closed circular. And again, the red strand closes on the red strand, the blue strand closes on the blue strand. Because they have opposite orientations, they can only close on themselves. So you get the topology of two interlinked circles. And so that's what DNA is. However, with DNA, it's generally a fairly long molecule. So when you close it into a circle, you can close it with various linking numbers. I could close, if this were a sort of an analog of DNA, its unstressed shape is untwisted, whereas DNA has a certain twist to it. So I can close it with that shape, and I get a sort of a relaxed structure that just, it's as relaxed as it feels like it can be. Or I can close it with a different linking number. I can put in a linking number of 1, or a linking number of 2, or any linking number I want. And the more link I put in, the more snarled up the DNA becomes. I'll explain a bit about that in a little while. But if you put in a linking number that is different than the relaxed value, then the DNA tends to have to snarl up one way or another. It tends to form these structures called supercoils. I'm sorry, this is a jargon word. I want to try to avoid jargon words, but it's a level of coiling where the whole double helix is coiling on itself. So it's a level of coiling above the coiling of the double helix itself, of one strand around the other. And that's why it's supercoiling, because it's a higher level of coiling. OK. And what makes this useful is that, first of all, you can see these as things become more, this is a relaxed molecule. As it becomes more supercoiled, it gets more tightly snarled up. And actually, more importantly, there are ways of separating these things chemically. So DNA is an acid. And an acid is a molecule that has an absent-minded hydrogen on it that will, in solution, go wandering off as a free proton, taking a positive charge away and leaving a negative charge on the rest of the molecule. So DNA, under solution conditions, will have a charge to it. So if you put it in an electrical gradient, it will run away from the negative pole and run towards the positive pole. So it will migrate in this direction. So what this is a gel. It's like Jell-O. It's a slab of gel. It's not made of gelatin. It's made of something else. But you put the DNA, your sample in on top, and then you just run an electric field, and it runs down the electric field. These are all molecules that are identical. They only differ in linking number. They have the same base sequence, the same everything, just the same length. They just differ in linking number. And if you get your gel conditions right, you can sort them out by linking number. So this band differs from that one by 1 in the linking number. And then you can take your little scissors and you can cut this band out, and you have a sample of DNA that is homogeneous in linking number. Every molecule has the same linking number. So you can do all kinds of experiments on it. Again, this is sort of the basic physical chemistry, a physical chemistry aspect of what I'm talking about here. OK, now the linking number is a controlled quantity. It's a topological invariant. So as long as you don't change the topology, the linking number is the same. So if you want to change the linking number, you have to either cut or glue. You have to do one of these forbidden operations on the molecule. And there are a lot of enzymes, which are little molecular machines that do specific jobs, that do these sorts of things. This is one that's called DNA gyrase. There's a T segment and a G segment here. These are both the double helix. Don't be confused. These are not the 2 strands of one double helix. This is a double helix, and this is a separate double helix. It's like, over here and over there on this belt. And so what this enzyme does is it grabs this G strand and the T strand, and it cuts the G strand, and it pushes the T strand through it, and then it closes it up again. So it does a double strand passage. Now, let me do that with this so you can see what happens. If you start out with this thing relaxed, and now I'm going to just push something through this, see, these are not really together, so I can push something through the gap between them. So I'll just take this thing and push it through the gap and close it up. And it has linking number 2. See, 2 full turns. So every time you do a double strand passage like this, you change the linking number by 2. So there are enzymes whose function is to regulate the linking number of DNA. Okay? Now, these can do a bunch of other things too in terms of the topology. If you have an unknot, something that is not knotted, a closed circle that's not knotted is called an unknot in mathematics, then you can push it around to get any geometry you want. All of these are still unknots. But now if you do a strand passage so that you take the one that's behind and you push it so now it's in front, then you can turn it into a trefoil. Or if you have a trefoil knot and you do a strand passage at any one of these 3, then you will unknot it. Okay? So the gyrase can change the knot type. It can knot and unknot DNA also. And again, in all of these, this rod-like thing is the double helix itself. And again, it can link and unlink. If you have two DNAs that are linked together, then by a strand passage, you can unlink them. And this is actually thought to be one of the major jobs of gyrase in the cell. But it can do all 3 of these jobs. And the other major job is that it controls the linking number. And that's the one that we're going to be focused on today. Now, okay, so that's some biology and some mathematics of the linking. Now a little bit of physics or physical chemistry. So DNA looks like a spring, and actually it behaves like a spring. It's got an unstressed twist rate, but you can crank it up or you can untwist it. Those deformations require energy. It wants to be approximately straight, but you can bend it, and that requires energy too. So it's like a spring. You can make all kinds of deformations of it, but it requires a bending energy to bend it and a twisting energy to twist it. And this is kind of its lowest energy shape that it wants to be. So now you can do mechanics, you can do engineering with this molecule. And so basically, a supercoiled structure like this guy has a higher energy than a relaxed structure because there's more bending involved here. You can see that it would be a higher energy. So here's a nice problem in basically molecular engineering. So you have a DNA molecule that's circular. It's closed with a fixed linking number, whatever it may be. I'll put in a linking number of 2 here, just for the sake of argument. And the question is, and now you have this molecule that is now elastic. It takes energy to bend and energy to twist. And it does take some energy to bend and twist this belt. It's not terribly elastic, but it's sort of enough for the purpose of illustration. And so the question is, now you've got a fixed linking number on something that can elastically deform, that requires energy to deform. So what kind of equilibrium configurations can it take on? And so that's a physics problem, or if you wish, an engineering problem. It's been solved. And the answers are that it takes on structures that are, these are the stable equilibrium configurations as you change the linking number. In this particular instance, you need to think of, I mean, what they're really doing is they're taking a molecule, and at the ends, they're putting in a little dial, and they're twisting this one relative to that one, so that when it gets to an integer, then you can close it, and it's a real linking number. But as long as you are just sort of thinking of this as something where you can dial in a parameter, a twist parameter here, then you can change the linking number effectively continuously. And that's what they're doing in this case. Okay, DNA has another trick. Back to some chemistry and physical chemistry. Any base pairs that form can separate. Now, you remember the AT base pairs had 2 hydrogen bonds and the GC base pairs had 3. Well, partly because of that and partly because of other reasons, it's easier to separate the AT base pairs than the GC ones. But nevertheless, anything that can form can separate. And the energy that it takes to do that is going to depend on what the base pairs are that you're trying to separate. Now, this is a critical function for DNA. DNA has two basic jobs. It needs to express genes. And to do that, what happens is that you have this complex, it's called a polymerase, which runs around, and it makes a messenger RNA, which is the red thing here, with the sequence that's complementary to one strand of the DNA. And the other strand has to get out of the way to allow this process to happen. So in order to initiate this process, you have to make a bubble where the DNA is strand separated. The other major job of DNA is that every time cells divide, they have to get a complete set of genetic instructions. So the whole of every chromosome has to make a copy of itself, and the copies have to segregate to the daughter cell. So every cell division, the DNA has to make copies of itself. Now, each strand has the information to synthesize the complementary strand. So all you do is separate the two, and then each one serves as a template for making the other strand, and there you are. That's basically how replication happens. But again, to initiate this, you have to pull the strands apart. So both of the major jobs of DNA require bubbles of strand opening to happen. So where and when these strand separations occur is very, very carefully controlled in biology. Now, back to the topology, how this can happen, how topology can drive this process. So here's a situation where you have a molecule that has 36 turns of DNA. They're numbered. This is 35, and the last one is 36. And and it's closed with a linking number of 32. So there are 4 of these supercoils, negative supercoils in this molecule. And this is a highly bent structure. It requires a fair amount of energy to form. There's another way, though, that you can package the thing with the same linking number. What you could do is that you could just open 4 turns of the double helix. So now you have a strand-separated region of 4 turns. So you have 32 turns that are still double helix, and the linking number is 32. So everything's fine. There's no residual deformation like this. Okay? So these are 2 ways that you can package the same DNA with the same linking number. And of course, you could do intermediates between these. You could open less and have some fraction of supercoils happening too. But these are just illustrations of the way that this can behave. Now, there's a cost to this transition. It costs energy to open these base pairs. But on the other hand, you get a benefit too, because you're relaxing the high energy of this highly deformed curved structure. So if the benefit exceeds the cost, then this will be the structure that will be preferred. And in that way, a deficiency of the linking number, that it's only 32, whereas to be relaxed it would have to be 36, can drive structural transitions, which can be involved with opening the DNA and giving access so that genes can express, so that replication can start. All these things can happen. And it's driven in some measure by the topology, what the linking number is. Okay, now back to some physics. You can calculate these things. You can take a DNA of thousands of base pairs long, put on any linking number you want. So now this is a molecule whose actual linking number is less than its relaxed value by 29 turns. It's under-twisted by 29 turns. If I made it, into something like this, it would have 29 turns instead of 2. And you can calculate what's the probability of strand separation of every base pair in it and get results like this. And the answers are actually quite good. When you do experiments to measure it, the open regions are experimentally found to be there and there, which is exactly where the theory calculates them to be. So again, this is physics. It's statistical mechanics. I'm not going to go into it, but just beyond just saying that this is a calculation also that one can do. Okay, back to DNA structure. This is not the only thing that DNA can do, this strand separation. It can also go into a left-handed helix. This is, you see this is winding in a left-handed way. The handedness of a helix, you can see this as left-handed, you can see this strand as going from back to front, and going to the left, okay? And this is my left hand, so that makes it left-handed. So you have right-handed helix, so the normal B form is a right-handed helix. This is a left-handed helix. And it requires a certain base sequence to form. It doesn't form in every sequence, but it's another thing that DNA can do. DNA has a surprising number of different types of structures it can have. This is a cruciform. In this, you see this particular strand goes up and back down. So this is a strand that is base pairing with itself. Now, in order to do that, you know, because A is paired with T and G with C, there's got to be symmetry to the sequence along this. So there are symmetry requirements that permit a cruciform like this to happen. This structure, in order to go, this strand-separated region, it's going from the helicity of a double helix to untwisted. So it's changing the twist. This guy, if you drive a transition to this structure, you're going from right-handed to left-handed. So you're doing more than just untwisting it, you're twisting it in the opposite direction. This one is also untwisted. This strand is a Watson-Crick structure, but it's base pairing with itself. It's not twisting around the other strand. So the helical twist is actually 0 for this. So both of these, so this one is kind of like this, that when you put the DNA into this structure, you are untwisting the double helix. And so the same considerations of going from a highly supercoiled, highly energetic one to a more relaxed DNA with this alternate structure in it, the same considerations, the same analysis happens for all of these types of transitions. And they have base sequence requirements to form. So you can also analyze these competitive transitions. And they are competitive, you see, because if one transition happens, if you have a molecule that can have different things happening in different places, and you put on some undertwist, then say it strands separate here, well, that packages the undertwist as that strand opening, and like that slide showed, it allows the rest of the sequence to relax somewhat. And so that means that the other transitions are less likely to happen, that every base pair sees what any other base pair is doing. So these are competitive transitions. So this is a sample calculation we did. I thought I'd show you it and explain it because it was on the poster for this talk. But this is an artificial sequence that has one region that's susceptible to strand opening, one very AT-rich region, one region that's susceptible to Z-form transition, and one region that's susceptible to cruciform. And then this is the linking difference. This is the actual linking number. And the actual linking number is here. The relaxed linking number is there. If the actual one is smaller than the relaxed one, so the whole thing is undertwisted, then this is a positive number, and this is the fraction of the relaxed linking number by which it's undertwisted. So this is sort of a fractional undertwist, is this number. So we looked at this for different cruciform sizes, and what we found is that, and this is as you supercoil this molecule more and more, as you change its topology by changing the linking number. So first of all, now, oh yeah, I should explain this. So cruciform transitions are green. Melting, which is strand separation, is red. zDNA, a little hard to read here, is blue. If you have the zDNA and the melting going both, then you get this magenta. If you have melting and cruciform both, then you get yellow. If you have zDNA and cruciform, then you get this teal green. And if you have all three happening, then you get white. Okay? So if you start out with a short cruciform, then first of all, there's nothing. At a certain stress level, in this particular example, the zDNA transforms. So now you have, at stress levels in that range, now you have only zDNA. And then a curious thing happens. So now you have this molecule, and a zDNA has popped out here. If you stress it more, a curious thing happens. The zDNA falls back, and the molecules and the strand separation happens. So you can get coordinated reversions and transitions where the zDNA reverting back to B form is coupled to the melting of a separate region at a different part of the molecule. And then, if you stress it even more, then the Z region comes back and you have them both happening, the melting and the Z simultaneously. Now, and that goes on, and if the cruciform is short, then it doesn't play any role at all. If the cruciform's a little longer, then eventually the cruciform also forms when you get stressed beyond a certain amount, and now you have all three transitions happening. If the cruciform is still longer, then suddenly it changes, the behavior changes, and suddenly the first thing that happens is this cruciform forms, and then you get the cruciform and then you get the same pattern here. You get the cruciform and the Z form, both of them, and then the Z falls back to B form, and you get the cruciform and melting. That's the yellow. And then beyond that, you get all three. So there's a very intricate dance that happens with the DNA, with these structural transitions happening here and there and everywhere, and reversions here coupled to transitions there. It's a very dynamic process, a very dynamic dance. And I don't want to go into this in too much detail, but when we analyzed 14,000 human genes at the stress levels that they occur in living cells, every single one of them had regions of alternate structure in them. These regions of alternate structure are basically universal at the stress levels that we have. Now, so far we've only talked about circles of DNA, and that sounds like a very specialized kind of thing to do. But in fact, this topological constraint is basically universal. Bacterial chromosomes usually are circular, but they are divided up into smaller domains by being held at the base. The chromosomes of higher organisms, like ourselves, also are held in a sequence of loops. Now, that is actually the same constraint as the constraint of circularity. It's effectively the same thing. I mean, if I put in, let me do it this way. If I put in, say, 2 turns, linking number of 2 here, again, okay? That constraint is the same, and it's really circular, as if I just turn this around and hold it at the base. It's the same thing. It's still got the same linking number. It's entirely the same. As long as I hold it so it can't twist at the base, then the constraint on a loop is topologically equivalent to the constraint of being circular. And all I would have to do to go from one to the other in this example is just pop it around and close it. Okay? But what's basically happening is exactly the same. So this constraint, this topological constraint, happens in all DNA all the time, in every cell, in every organism. Okay, now there's another way in which the topology can be, in some sense, changed. I talked about these enzymes, such as gyrase, that do strand passages and change the topology. But there's another way in which this can effectively happen, which is that when the genes are expressing, they're expressing by this polymerase moving along and making an RNA. Remember that. But what happens is that as this complex pushes along, it pushes over twist ahead and it leaves under twist behind. So for example, in this case, I don't know whether you'll be able to see this. So if I put in a turn here, and then I start doing something that involves strand separating so that the downstairs piece is untwisted, what I'm effectively doing as I creep along is I'm pushing the twist into the upstairs piece, and it's getting higher and higher twist on it, and the downstairs one is relaxed. So you can think of this 2 ways. This is sort of like a moving domain boundary between a highly stressed region and an unstressed region, or you can think of it as just repartitioning the twist to the region it's not yet gotten to. But this is basically what the polymerase is doing. Now, yeah, this is just another slide that sort of shows it, that what's happening is that downstream from the polymerase, the region it's already passed through is strand separated still, and so it's untwisted relative to the upper region. So it's pushing over twist ahead and leaving a wake of undertwist behind it. Okay, so I want to tell you a story, a biological story in the end that kind of puts all of this together so that you can see how it plays out. This is a story about the c-Myc gene. c-Myc is a particular gene. It makes a protein called the c-Myc protein, which is something called a growth factor. So it affects how cells grow. And it's a very important gene in mammalian biology. It's called an oncogene. Onco means cancer. And oncogenes are genes where when you misregulate them, they cause cancer. This particular one, c-Myc, causes a cancer called Burkitt's lymphoma, and it's also involved in a whole bunch of other types of cancer. The c-Myc protein that this gene makes, like I say, it's a growth factor. If you get too much of it in the cell, then the cell grows in an uncontrolled way, and that's the cancer. If you get too little of it in the cell, then the cell stops growing, and it senesces, and eventually it will die. So you have to get this just right, you know, not too hot, not too cold, not too much of this protein, not too little of this protein. It has to be just at the right concentration and maintained at the right concentration within a factor of about 4 or 5 all the time. Now, for reasons that I can't explain, and one doesn't really look for explanations in biology, this c-Myc protein is rapidly degraded. So, in fact, as it's made, it has a short half-life in the cell. So how much it is there, the abundance of it, is primarily determined by how much it is expressed. So the rate at which its gene is expressed is the determining factor, the major controller for how much of this stuff is in the cell. And it has to be regulated rather carefully that way. So these P's are the various sites. These 3 are the major sites where this gene expression starts. Okay? Now remember that as it expresses, it pushes over twist ahead, and it leaves a wake of undertwist behind it. Now there are these various regions, Z1, Z2, Z3, and in particular this fuse region. This is called fuse far upstream sequence element. That's why it's called fuse. It's about 150, it's about 1,500 base pairs upstream. These little marks are 500 base pairs apart. So it's about 1,500 base pairs upstream from where the gene tends to start. Okay? And it is a region that easily strand separates. So when the transcription, when the gene expression, when the polymerase moving along pushes under twist behind, it opens this region. It causes it to strand separate. Now the way it does it is actually bimodal. This is the linking difference going from -12 to -20. Well, expressed this way, it's from 12 to 20 because the actual linking number is smaller than the relaxed value by that much. And you find that one region opens very quickly and the other region opens relatively slowly. So one region opens at relatively lower stress levels, the other region opens at higher stress levels. So an early melting region and a late melting region. Now, there's a protein that binds to this fuse element. And that protein regulates the next round of transcription. When it binds to the early melting region, and it only, this protein, I should say, only binds to single-stranded DNA. If the DNA is in a duplex, it cannot bind. If the DNA wants to go back from single-stranded to duplex, the protein falls off. Okay? So this protein binds to the strand-separated piece of the DNA. When it binds to the early melting region, it initiates the next round of transcription. When it binds to the late melting region, it inhibits the next round of transcription. So binding only here, that will cause another round of transcription to start. Binding here will cause it to not start. It will prevent it from starting. And if it binds to both, which commonly happens, then the inhibition dominates. Okay, so the late melting region dominates over the early melting one. Okay, so what happens is this. This gene starts expressing. It pumps negative supercoils back, under twist back. It causes this region to become more stressed. It causes it to have a smaller linking number. And that causes the late melting region to open. And then the protein binds to the late melting region, and it inhibits the next round of transcription. The next round of gene expression, sorry. And so the gene expression, the polymerase pushes along and along and along, and it's pushing negative supercoils, negative twist back the whole time. But eventually it gets beyond this point. And at that point, now it's pushing negative supercoils, it's pushing under twist back here. And now this is performing another transition. The negative supercoils are going to driving a different transition. So they don't get past this point to keep the fuse element open. So the negative supercoils that are over here, now they're, this is a dynamic process. These are like waves of twist passing down the DNA. The waves that were here are, they pass along and the DNA becomes progressively relaxed, whereas the waves that are being produced now by the polymerase are going to opening this, to causing a structural transition in this region. So this one relaxes somewhat, and that means that the late melting region falls back to B form, turns back to B form, and the protein ceases to bind to that region, leaving it only bound to the early melting region, and that activates the next round of transcription. So this whole thing is a gate. It's a topological gate is what it is. So you start a round of transcription, and by the structural transition 1,500 base pairs away, you inhibit the next round of transcription until the last one has gotten past here and gotten past it far enough so that this guy has fallen back enough so that the next round runs. So so once the polymerase gets perhaps up to about here, then the next round happens. And so you get rounds of transcription, rounds of gene expression, one after another, paced, gated by this particular specific distance. And that is how this gene is regulated so that it expresses just at the level it needs, not too much, not too little, to make just the right amount of the c-Myc protein to keep everything right for the cell. And it's a topological gate. It's determined by the linking characteristics of this particular region. And it's determined by, it involves waves of undertwist that pass down the DNA, structural transitions that happen in the DNA, it's a very, very active process. So this view of DNA that I want the biologists to, in the group especially, to bring back is that it's a highly dynamic molecule and it really actively participates in the mechanisms by which it's regulated. It's topologically constrained, and the level of the topological constraint, the linking number, is closely regulated by a bunch of processes. And its activities are determined by how topologically, what its topology is, how it's stressed by the topology. As genes express, there's waves of twist that pass down the molecule. These can drive structural transitions, which can affect regulation and mechanisms of regulation in a variety of ways. The one I showed is only one that involved the binding of a protein to these alternate structures. But there are many other ways that these things can and do happen. And of course, these things can be modulated by a variety of events. So for example, one thing that I didn't talk about, if you have this DNA and it's got these twist waves driving down it dynamically, as long as the DNA is fairly straight, it can twist. But if you bind a protein to it that binds and bends the DNA, suddenly the twist up here gets transformed into pulling this big lever arm through a solvent, a solution, which is very viscous at this size scale. And so suddenly everything changes. The stress won't be able to get down beyond this point very far. And so suddenly all the constraints change. So you need to think about DNA as a very active, very dynamic molecule with all kinds of ways in which binding events and structural transitions can affect its activity. It's a very different picture than the sort of passive view of DNA that people have. So this is a picture in the end that kind of brings everything together. But I wanted to show you an example of how, especially in biology, especially in modern biology, you need to understand mathematics, you need to understand physics, you need to understand chemistry to understand these biological processes. And these, although, like I say, at certain stages in one's education, one studies these things as separate disciplines. In fact, in the real world, they come together and merge together, and you need them all to understand a lot of the things that happen in living systems especially. So, well, thank you. In this fuse, one of the things that makes biology very difficult is that every story is its own story. This fuse element is something that is specific to the c-Myc gene. There are many, many other stories where structural transitions play roles in regulation. But it's very difficult to actually tease out the details at this level of what's actually happening biologically. And so that kind of research is really one gene at a time. So this has only been fully explored in the c-Myc case, and motivated by the fact that c-Myc is so important because of its cancer-causing characters.